Dear student, this is Dr. Prakash Mungli, Professor of Biochemistry. So in this glycogen metabolism lecture, I will be going over glycogen synthesis and glycogen degradation. Let's get into the details. Glycogen, it is a storage form of glucose. So whenever person has a sufficient glucose in the blood, and when the blood glucose is entering into a liver and skeletal muscle, and when the liver and skeletal muscle and all other tissues, when they have sufficient glucose for glycolysis and for the energy purpose, so the additional glucose that is available will be diverted into a glycogen molecule formation, and this glycogen is stored in the tissue and it will be used for energy purpose or the purpose of maintaining blood glucose whenever person goes into fasting condition, or whenever the skeletal muscle needs energy or the tissue needs energy. Now we are taking into consideration two major organs or two major tissues here to go over glycogen metabolism. One is liver, the other is skeletal muscle. The reason for that is liver glycogen stores is around 5% of its weight, whereas skeletal muscle glycogen stores is around 0.7% of its weight. But note that although percentage of liver glycogen is 5% of tissue weight, but the tissue weight is 1.8 kgs only. That's why the content of glycogen in the liver is 90 grams. Whereas look at the muscle glycogen, although the percentage of glycogen present in the muscle is only 0.7% of its weight, but the weight of skeletal muscle in a 70 kg man is 35 kgs. So accordingly, the body content of glycogen in the skeletal muscle is 245 grams. That means skeletal muscle simply contains much higher quantity of glycogen compared to the liver glycogen. And also the extracellular glycogen that is present is around 10 grams. Now the purpose of liver glycogen is to maintain the blood glucose level, whereas the purpose of skeletal muscle glycogen is to maintain the energy needs of, whereas the purpose of skeletal muscle glycogen is for its own use. Now let's look at glycogen structure. Glycogen is present in the cytoplasm as glycogen granules, and the glycogen is a highly branched polymer of glucose, and the linear chain of glycogen, it is made with the alpha-1,4 glycosidic bonds, where one glucose is linked with another glucose with the alpha-1,4 glycosidic bond, meaning the first carbon of a glucose is going to interact with the fourth carbon of an incoming glucose with the alpha-1,4 glycosidic bond, and that makes a linear chain. And also glycogen, since it's a highly branched polymer, there will be a branch point at every 8 to 10 glucose residue. So that branch point is made because of alpha-1,6 glycosidic bond. So make sure you remember that. So the linear chain is made with alpha-1,4 glycosidic bond, whereas the branch point is made with alpha-1,6 glycosidic bond. Now here is the structure of glycogen granule. So as you see here, so the linear form of glycogen structure, it is made with alpha-1,4 bond, whereas the branch point, wherever you see the branch point, only at the branch point, so there will be alpha-1,6 bond. Let me just write down the structure and show you how exactly this alpha-1,4 and alpha-1,6 bonds are made here. So I'll just write a general structure, one glucose molecule, and the other glucose molecule here. So here is, this is the first carbon of one glucose, fourth carbon of the other glucose here. So there will be formation of alpha 1,4 glycosidic bond with the first glucose and the incoming glucose, that's the second glucose. Now like this, a linear chain will be synthesized as more and more glucose molecules make a connection like this, right? So how is the alpha 16 branch point is created? Just to show you that, so the numbering in the carbon atom is, say this is number 1 carbon, number 2, number 3, number 4, number 5 and number 6 carbon. And now the sixth carbon here, it is going to interact with the first carbon of incoming glucose molecule. So the first carbon, I am going to write another glucose molecule up here. Now here is the first carbon, second, third, fourth, fifth and sixth carbon. So the bond linkage here is the sixth carbon is interacting with the first carbon, making a linkage there. So this particular linkage is alpha 16 glycosidic bond. And this is the branch point that a glycogen will have. So like this, a glycogen is a highly branched polymer of glucose. Alright, so every glycogen, as you see, look into the structure, so every glycogen has got a protein in the center and that is a glycogenin. So there will be a glycogenin in every glycogen molecule, and that glycogenin, from that glycogenin, so glycogen structure will be branched out and a highly branched glycogen will be synthesized. So that means glycogen is not a pure carbohydrate. So there will be a protein in the center of glycogen molecule, and we are going to see that as we move on to see glycogen synthesis. A protein glycogenin is in the center and interacts with the reducing end of glucose, and in the periphery, whatever the glucose molecules that you see here in the periphery of glucose, at the end of every branch of glycogen, all these peripheries, they are basically non-reducing ends. So there is no reducing end in a glycogen molecule, so there will be only non-reducing ends. All the branches have non-reducing ends, and there will be multiple branch points with non-reducing ends will be present. And the advantage of having so many branch points which are having a non-reducing ends here is it will allow a rapid degradation of glycogen and also rapid synthesis of glycogen whenever the glycogen is partially degraded, because all it takes is, in the rapid degradation, all the glycogen degradation enzymes can attack all these branches simultaneously, and thereby glucose molecules are released at a time. And also when partially degraded glycogen is there, so restoration of that glycogen, like remaking that glycogen back to the normal form, can be done easily, because all these branch points can be simply elongated to bring it back to the original structure. So the rapid degradation and rapid restoration of glycogen is made possible only by having higher number of branch points. Each glycogen granules, for that matter, contains about 30,000 glucose residues, and this number can vary from 2,000 to 60,000, but on an average there will be around 30,000 glucose residues in each glycogen granule which is present in the cytoplasm, and these glycogen granules are highly branched polymer of glucose, and having a high branches, so many branches will help in degradation, rapid degradation and rapid restoration of glycogen. Now coming into glycogen synthesis. In the glycogen synthesis, certain enzymes participate in the synthesis of glycogen. So whenever a glucose is available plenty at that time, especially in the liver and other tissues, glucose is converted into glucose-6-phosphate done by glucokinase, hexokinase. So I'm going to write that here. So the conversion of glucose into glucose-6-phosphate, that is done by either glucokinase or hexokinase enzyme, as you all know by this time. So this is the enzyme that converts glucose into glucose-6-phosphate. Now glucose-6-phosphate will be converted into glucose-1-phosphate. That will be done by enzyme phosphoglucomutase. Now the glucose-1-phosphate will undergo a reaction that will be catalyzed by UDP-glucose pyrophosphorylase enzyme to become UDP-glucose. During this time, there will be the UTP coming into the reaction, getting out of the reaction as PPI. So uridine triphosphate gets into the reaction and it will get out of the reaction as inorganic pyrophosphate and making UDP glucose, and that reaction it is catalyzed by UDP glucose pyrophosphorylase enzyme. Now this pyrophosphate which is made here, that pyrophosphate will break into 2 inorganic phosphate molecule, 2 PPI, and that is catalyzed by enzyme pyrophosphatase. Okay, thereby exergonic reactions that are going on here, where uridine triphosphate it is releasing inorganic pyrophosphate and making UDP glucose. That's the exergonic reaction. And also the breakdown of pyrophosphate into 2 inorganic phosphate is also an exergonic reaction. So that means this entire system here is made exergonic, and that there will be a lot of energy available in the system to make a glycogen now. So to make glycogen, UDP glucose is necessary, and that is also prepared now. So the glucose converted to glucose-6-phosphate, glucose-6-phosphate converted to glucose-1-phosphate, glucose-1-phosphate is now converted to UDP glucose by using UTP, uridine triphosphate. And also UTP breakdown is exergonic and PPI breakdown is exergonic. Now the energy is there in the system. Now UDP glucose can go into glycogen formation, and initial glycogen formation is an autoglycosylation process where a glycogenin, it has got a tyrosine, amino acid tyrosine with the hydroxyl group. So UDP glucose will be attached to this hydroxyl group, and that's how a glycogenin molecule is attached with the glucose, and UDP will be released. This particular process do not need any enzyme mediated action. Glycogenin, which is a protein, itself act as an enzyme and add glucose over its surface, especially on the tyrosine residue. Like this, about 8 glucose residues will be added, 3 glucose plus. So basically 8 glucose residues will be added over glycogenin to make a glycogen primer. So the glycogen primer formation do not need any enzymatic action, do not need any enzyme. It is an autoglycosylation process. Once 8 glucose residues are made by autoglycosylation process, so that glycogen synthase enzyme will come. It is going to use UDP glucose coming from these reactions and elongate that particular chain for up to 11 glucose residues. Once it reaches 11 glucose residues, so a part of the chain is broken down. It will be transferred to other point in the glycogen chain, and that creates a branch point done by branching enzyme, which is called as amylo-4,6 transferase. So like this, glycogen synthesis will be going on in the cytoplasm whenever we are in well-fed condition. So some of the enzymes to recap again. So we have glucokinase hexokinase reaction. We have phosphoglucomutase enzyme converting glucose-6-phosphate into glucose-1-phosphate. UDP glucose pyrophosphorylase converting glucose-1-phosphate into UDP glucose. Pyrophosphatase enzyme breaking pyrophosphate into inorganic pyrophosphate, making system exergonic. Autoglycosylation process where glucose is added onto a tyrosine of glycogenin to make a glycogen primer, and then glycogen synthase coming and elongating the glycogen primer, and finally branching enzyme coming and introducing a branch point. Like this, a highly branched glycogen structure will be synthesized in the cytoplasm when a person is in well-fed condition, when glucose is sufficiently available. So here is the figure that will explain all the things that I have explained in the previous slide. So let's go one more time. So here is a glucose-6-phosphate coming from glucokinase and hexokinase reaction. This glucose-6-phosphate will be converted into glucose-1-phosphate done by phosphoglucomutase enzyme. Glucose-1-phosphate will be converted into UDP-glucose by breaking UTP and releasing PPI, and this PPI further breaks into 2-inorganic phosphate by pyrophosphatase enzyme. So that makes the system hexagonal. Now the UDP-glucose which is available here will go and attach with the hydroxyl group of tyrosine residue, and this attachment doesn't need any enzymatic action because glycogenin itself acts as an enzyme, and that process we call it as autoglycosylation. The addition of UDP glucose, where UDP is released, glucose is added, it is continued without any enzymatic action up to 8 glucose residues being added. Once 8 glucose residues are added, it is made a glycogen primer there, and this glycogen primer will be simply elongated by glycogen synthase by bringing more UDP glucose and adding glucose and releasing UDP every time. So all this linear chain synthesis, it is done by creating alpha-1,4 glycosidic bond. When around 11 glucose residues are there in this glycogen linear chain, so now the branching enzyme, it is going to take a portion of this particular linear chain and reattach it somewhere else. So that is what is shown here. As you can see in this particular figure, a partially degraded glycogen, it is shown here, where a glycogen synthase is elongated both the chains here. I am highlighting that. A partially degraded glycogen is elongated on either side of the chain with the red dot glucose residues that are shown here, and then a part of that, whenever sufficient elongation is done, a part of that is removed as shown by the dotted line, and it will be transferred to some other branch. Okay, that's how a new branch is created with alpha-1,6 glycosidic bond. So a branching enzyme is going to break alpha-1,4 bond. Here is the alpha-1,4 bond that it is breaking. It's going to break this alpha-1,4 bond, take this entire unit, and it is going to transfer that here and create alpha-1,6 bond there. So alpha-1,4 bond is broken. Alpha-1,4 bond is broken here and creating alpha-1,6 bond. And this type of bond formation is done by a branching enzyme, and that is why branching enzyme is also called as alpha-1,4 to alpha-1,6 transferase. It is also simply written as 4,6 transferase. So 4,6 transferase is a branching enzyme. Remember that. Why 4,6? 4,6 is because alpha-1,4 bond is clipped and it is creating alpha-1,6 bond here. Okay, and that's why the enzyme is called as 4,6 transferase or alpha-1,4 to alpha-1,6 transferase. Like this, with the help of glycogen synthase enzyme and the branching enzyme, a highly branched glycogen structure is made here. Okay, so that is how the glycogen synthesis is done. So let me recap one more time. So the glycogen synthesis, it begins with the conversion of glucose into glucose-6-phosphate and glucose-6-phosphate into glucose-1-phosphate, glucose-1-phosphate forming UDP-glucose, and this UDP-glucose is diverted into attachment of UDP glucose to glycogenin. Around 8 UDP glucose residues are getting into the reaction, only glucose is added, UDP released, and that process is catalyzed by glycogenin, and it's an autoglycosylation process. Once a sufficient linear chain is prepared, a part of the linear chain is clipped and then it will be transferred to some other point in the other chain and creating a alpha-1,6 glycosidic bond, and that's the branch point. That means a glycogen synthase enzyme which is elongating the glycogen primer chain, and the branching enzyme which is a 4,6 transferase or alpha-1,4 to alpha-1,6 transferase. With the help of these two enzymes, a highly branched glycogen is synthesized, and the branch points here, for every 8 to 10 glucose residue there will be one branch point in a glycogen molecule. Remember that. That's how a glycogen is synthesized. Now let's get into glycogen degradation. Whenever person is going into fasting condition, or whenever person is exercising, or whenever person is in stressful situation where epinephrine is released, at that time the glycogen that is stored in the liver, the glycogen stored in the skeletal muscle, or the glycogen present in other tissues, it can undergo degradation. Now during the glycogen degradation, there will be removal of glucose units by breaking alpha-1,4 glycosidic bond and alpha-1,6 glycosidic bond. Now how this is done? So glycogen degradation, it is initiated by enzyme glycogen phosphorylase. Note that the glycogen phosphorylase, as it is there in the name, it is going to incorporate inorganic phosphate as a nucleophile and break alpha-1,4 glycosidic bond and release glucose-1-phosphate molecule. And for glycogen phosphorylase, it needs pyridoxal phosphate as the coenzyme, and that will help in the phosphorylysis of alpha-1,4 glycosidic linkase by incorporating inorganic phosphate. Meaning, inorganic phosphate is necessary for the degradation of glycogen to release glucose-1-phosphate, and also pyridoxal phosphate is acting as a coenzyme. Make sure you remember this point. So if there is a deficiency of inorganic phosphate in the cytoplasm, that will decrease the action of glycogen phosphorylase, thereby glycogen degradation decreases, and that can lead to hypoglycemia. That is why in hereditary fructose intolerance, where there will be trapping of inorganic phosphate, or in classic galactosemia, where there is a trapping of inorganic phosphate leading to overall deficiency of inorganic phosphate, and that will lead to decrease in glycogen phosphorylase enzyme action. And when there is a decrease in glycogen phosphorylase enzyme action, that will decrease glycogen degradation. And when there is a decrease in glycogen degradation, especially in the liver, that will cause hypoglycemia. And that's why whenever there is a decreased availability of inorganic phosphate, so that will lead to decreased glycogen degradation, and thereby it will contribute to hypoglycemia in conditions like hereditary fructose intolerance, which is there in fructose metabolism, and classic galactosemia in galactose metabolism. Okay, these are some of the conditions or examples for disorders where there will be decrease in inorganic phosphate in the cytoplasm. Now, removal of glucose residue from non-reducing ends until 4 glucose residues remain at the branch point will be done by glycogen phosphorylase enzyme. Now, see, the glycogen phosphorylase enzyme, when it is active in the presence of glucagon, it is going on removing one glucose residue at a time by incorporating inorganic phosphate at the non-reducing end and releasing it as glucose-1-phosphate. Example, so inorganic phosphate is incorporated here and release this glucose as glucose-1-phosphate. Now it will go to the next glycosidic bond, alpha 1 for glycosidic bond, incorporate inorganic phosphate, release the second glucose as glucose-1-phosphate. Like this, it will go on incorporating inorganic phosphate, use it as a nucleophile and release glucose as glucose-1-phosphate. How long glycogen phosphorylase will do this? As long as there are 4 glucose residue left at the branch point. This is what it is. Look at this now. I'm going to color this or shade it. So now count the number of glucose here in this branch point. So on either side you have number 1, 2, 3 and 4 glucose. This side 1, 2, 3 and 4 glucose. So there are 4 glucose residue at the branch point and acting like a fork, right? So whenever you have a fork like thing, 4 glucose residue at the branch point, so it cannot go into the active site of glycogen phosphorylase and that causes steric hindrance, and that's when glycogen phosphorylase will stop degrading glycogen because of the steric hindrance. It cannot go into the active site of a glycogen phosphorylase. Now what will happen? So at this time comes debranching enzyme. Now the debranching enzyme, it is having 2 enzymatic subunits. One is 4,4-transferase component of debranching enzyme, which is called as glucan transferase. Other is alpha-1,6-glucosidase component of debranching enzyme. So debranching enzyme is a bifunctional enzyme. It has got 4,4-transferase function and it has alpha-1,6-glucosidase function. Now let's see what is the function of 4,4-transferase and what is the function of alpha-1,6-glucosidase. Now the 4,4-transferase, what it does is, it is going to transfer 3 glucose residues from one of the branch point to the other branch. Now how that happens? As it is shown here in the figure, I am going to shade that or highlight it. Here is the 3 glucose residues, right? So glucan transferase component of debranching enzyme, that is 4,4-transferase. Why it is called as 4,4-transferase? Let me explain that to you. So what it does, it is going to break this bond here, that is alpha-1,4 bond. It's going to break that, take this 3 glucose residue and transfer it here by creating alpha-1,4 bond. So basically a glucan transferase enzyme or the 4,4-transferase enzyme, what it does is going to cut that alpha-1,4 bond in one of the branch and take those 3 glucose residue and attach it to other branch. Okay, so thereby elongating the other branch by 3 more glucose. And that's what is shown here. As you can see, after the attachment, see here, 3 more glucose residues are added here, right? So basically the enzyme, it has clipped alpha-1,4 bond and reformed alpha-1,4 bond. That's why it is called as 4,4-transferase or alpha-1,4 to alpha-1,4 transferase. Okay, that's what is the 4,4-transferase part of debranching enzyme. Now there is only one glucose that is left at the branch point here after the action of 4,4-transferase. Let me highlight that. Here is this last glucose that is left at the branch point with alpha-1,6 bond. This is alpha-1,6 bond here. This particular branch point has a alpha-1,6 bond, and that alpha-1,6 bond has to be clipped by alpha-1,6-glucosidase enzyme. That is the other functional unit of debranching enzyme. So the debranching enzyme has 4,4-transferase, which will remove 3 glucose residue from one of the branch by cutting alpha-1,4 bond and reforming alpha-1,4 bond on the other side, elongating that chain, and the last glucose which is remaining there, it will be removed by alpha-1,6-glucosidase and it will be released directly as glucose, not as glucose-1-phosphate. That's a direct glucose that we get. Now what happened to this chain here, which is a long linear chain now, and this long linear chain that is remaining now, it will be taken care by the glycogen phosphorylase enzyme. Now the glycogen phosphorylase, it is going to incorporate inorganic phosphate one at a time and release glucose-1-phosphate molecule, and that's how glycogen is degraded. So if sufficient time is allowed, like if a person is fasting for almost to 24 hours and more than 24 hours, entire glycogen will be degraded all the way until glycogenin. That's a complete degradation of glycogen. But usually what happens on day-to-day basis, there will be only partial degradation of glycogen, and that partially degraded glycogen can be replenished, rebuilt again by glycogen synthase and branching enzyme. So like this, in glycogen degradation process, there are two enzymes which are involved here. One is glycogen phosphorylase enzyme, which is involved in the degradation of glycogen starting from the non-reducing end by introducing inorganic phosphate as a nucleophile and releasing glucose-1-phosphate at a time, and this will go on all the way until 4 glucose residues are left at the branch point. The fork-like thing there, because of the 4 glucose residues on either side, it will create a steric hindrance for glycogen phosphorylase, so the enzyme will stop catalyzing the reaction. And then at that time a debranching enzyme comes, and this debranching enzyme has two enzymatic subunits. One is 4,4 transferase. What it does is going to take only 3 glucose residue from one of the branch and reattach those 3 glucose in the other branch and elongating it, right? Now the last glucose that is left over with alpha-1,6 bond there, it will be clipped by alpha-1,6 glucosidase and release that as directly glucose. Now the glycogen phosphorylase will come and take out 1 glucose at a time from that long linear remaining glycogen structure, and that's how glucose-1-phosphate will be released. Now how many glucose-1-phosphate will be released per glucose that is released by alpha-1,6 glucosidase? It is for every 1 glucose that is released, there will be 7 to 9 glucose-1-phosphate will be released. So glucose to glucose-1-phosphate is 1:7 to 9. Okay, so remember that number, because there will be 1 branch point for every 8 to 10 glucose residue. A branch point, there will be 1 branch point for every 8 to 10 glucose residue. That means 1 glucose will be released for every 7 to 9 glucose-1-phosphate released by the action of glycogen phosphorylase and debranching enzyme. Now what happened to the glucose-1-phosphate that is released by glycogen phosphorylase? Because out of every 7 to 9 glucose residue which is released as glucose-1-phosphate, there is only 1 glucose that is released. So majority of the molecule that is coming out of glycogen degradation is glucose-1-phosphate only. So this glucose-1-phosphate, as it is shown here in the figure, glucose-1-phosphate will be converted into glucose-6-phosphate by phosphoglucomutase enzyme, and the glucose-6-phosphate further it will be converted into glucose, and that is done by glucose-6-phosphatase enzyme. Note that glucose-6-phosphatase enzyme is located in the membrane of endoplasmic reticulum, and it is seen in the liver predominantly, kidney, cortex, small intestine and beta cells of pancreas. So there will be 2 to 3 fold increased expression of glucose-6-phosphatase enzyme during starvation and in diabetes mellitus. And also liver glycogen maintains blood glucose level during fasting, because the glycogen degradation that is going on in the liver, the purpose of that is to make it as glucose, and glucose is released into the bloodstream to maintain the blood glucose. That's the purpose of glycogen that is present in the liver. Now the glycogen degradation in the skeletal muscle and other tissues, it differs from the hepatic tissue glycogen degradation, because other tissues like skeletal muscle and extrahepatic tissues other than the liver, kidney, beta cells of pancreas and small intestine, so other tissues they do not express glucose-6-phosphatase enzyme. So what they will do with the glycogen degradation? As it is shown here in the figure, glycogen which is broken down into glucose-1-phosphate will be converted into glucose-6-phosphate, and this glucose-6-phosphate in these tissues will enter into glycolysis. Now it all depends on which type of glycolysis is going on in the tissue. Example, in the muscle, in the white muscle fibers, which are referred as fast twitch muscle fibers, where they contain less mitochondria, so majority of it is going into anaerobic glycolysis, making lactate as the end product. In a red muscle fiber, that is slow twitch muscle fibers, glucose-6-phosphate enters into aerobic glycolysis and making carbon dioxide and lot of ATP is given. Now the glycogen in these tissues are used as an emergency fuel source for energy, not to maintain the blood glucose level. So the glycogen, the purpose of glycogen present in the skeletal muscle is for its own use, not to make a glucose or maintain the blood glucose level. Now this figure is giving an overview of both the pathways. On left hand side you are seeing a glycogen synthesis, on the right hand side you are seeing a glycogen degradation. Now the glucose, excess glucose getting into glucokinase reaction, making glucose-6-phosphate in the liver, and in other tissues it is done by hexokinase. Glucose-6-phosphate converted into glucose-1-phosphate. Now the UDP-glucose pyrophosphorylase making UDP-glucose. UDP-glucose getting into glycogen synthase reaction or glycogen primer formation by autoglycosylation process, and later glycogen primer entering into glycogen synthase reaction, where more and more alpha-1,4 glycosidic bonds are created, and once sufficient lengthening is done, branching enzyme is coming in and introduces a branch point. So the simultaneously glycogen synthase and branching enzyme, it works in a coordinated way to generate highly branched glycogen granules and in the cytoplasm. Now insulin is going to promote the action of glycogen synthase, whereas glucagon is going to decrease the action of glycogen synthase. Now, when the person goes into fasting condition, glucagon is increased and it is going to promote glycogen degradation, whereas insulin is decreased. That means glycogen synthesis decreases, glycogen degradation increases. So now the glycogen phosphorylase, what it does, it uses inorganic phosphate as a nucleophile and release glucose as glucose-1-phosphate, and all the way until 4 glucose residues are remaining at the branch point. When 4 glucose residues are remaining at the branch point, now comes the debranching enzyme, and the debranching enzyme has 2 enzymatic subunits. One is a glucon transferase enzyme, which is 4,4 transferase, other is alpha-1,6 glucosidase. Now the glucon transferase, what it does, it is going to take 3 glucose residue out of one of the branch point and reattach it to other branch point. And the last glucose residue which is there at the branch point with the alpha-1,6 linkase is clipped by alpha-1,6 glucosidase and release that as a free glucose molecule. Now the glucose-1-phosphate that is released by glycogen phosphorylase enzyme, it will be converted into glucose-6-phosphate, and that will be converted into glucose in the liver, or it will just go into glycolysis in other tissues. And this is what is the overview of glycogen degradation in the liver, skeletal muscle and other tissues, glycogen synthesis and glycogen degradation in fact, and this happens in the cytoplasm. Now let's move on to see glycogen degradation in the lysosomes. Around 1 to 3% of glycogen is continuously degraded in the lysosome, because we have lysosomal hydrolases, and one of the lysosomal hydrolase that will degrade glycogen in the lysosome is acid maltase enzyme. This acid maltase enzyme, it's a combination of 2 enzymes, that is alpha-1,4 glucosidase enzyme and alpha-1,6 glucosidase enzyme. Both are acid hydrolases, which will degrade glycogen in the lysosome. So the function of alpha-1,4 glucosidase is to break alpha-1,4 glycosidic bond. And by breaking alpha-1,4 glycosidic bond, it is going to release glucose there. The function of alpha-1,6 glycosidase is to break alpha-1,6 glycosidic bond, and that will also release glucose. So basically, glycogen degradation in the lysosome using alpha-1,4 glucosidase and alpha-1,6 glucosidase, both are acid stable enzymes. Together they are called as acid maltase. So they are directly release glucose from glycogen, as opposed to glycogen degradation that is going on in the cytoplasm catalyzed by glycogen phosphorylase, which is breaking glycogen and releasing glucose-1-phosphate, not glucose. So here in the lysosomal glycogen degradation, glucose is released, not glucose-1-phosphate. So deficiency of acid maltase enzyme, it will give rise to a disease called Pompe disease. Pompe's disease is a glycogen storage disorder in the lysosome. So that is about glycogen metabolism, where I have explained you how glycogen is synthesized and how glycogen degraded and all the enzymes that are necessary for the synthesis of glycogen and degradation of glycogen. I hope you understood this particular important concept of glycogen metabolism. Thank you.